Well, good afternoon, everyone. It's so wonderful to be with all of you. And welcome to Back to the Future of Organ Transplants, the latest stop in our virtual expedition series, where we take you behind the scenes of the University of Alberta Hospital site and introduce you to some of the incredible clinicians and researchers who are doing amazing things right here in Edmonton. My name is Carolyn Thompson, and I'm the Director of Philanthropy at the University Hospital Foundation. I would like to first acknowledge that we are located on Treaty 6 territory and respect the histories, languages, and cultures of First Nations, Métis, Inuit, and all First People of Canada, whose presence continue to enrich our vibrant community. Today's session is being recorded, and we will send everyone the link who responded to our invitation over the next few days. As you watch the presentations and your questions arise, we invite you to submit them through the Q&A button at the bottom of your screen. A Q&A session will follow the presentations where your questions will be answered. Now, before we introduce our speaker, I would like to take a moment to tell you about who the University Hospital Foundation is and what we do, and most importantly, why we do it. We believe that life is better when you are healthy. As agents of hope, we strive to match the determination and intensity of the medical teams we support, including Canada's most comprehensive transplant program at the University of Alberta Hospital, and the only program that performs the full range of surgeries, including heart, heart-lung, double lung, and islet cell transplants. Over the past 10 years, generosity from individuals and corporate partners have contributed nearly $220 million towards bringing some of the best care in the world to our communities, and likewise, sharing medical discoveries from innovators with the world. Through our donors, we provide ongoing financial support to advance patient care, create new knowledge, and make breakthrough discoveries at one of the leading academic medical centers in Western Canada. And now we would like to learn a little bit more about how you are connected to transplant care. We invite you to participate in a live survey. Please select the one question that best represents why you decided to join us for today's expedition.
The results are starting to come in. We will give everyone about 10 more seconds.
Now I do have to put my glasses on for this. So it looks like we have a range of reasons why people are joining us today. Some who have had an organ transplant, or some that are a caregiver to someone who has had the organ transplant. And even looks like we have some students and researchers with us today and some of our corporate partners. But most of you are just interested in learning more about organ transplantation. So no matter what brought you here today, we're so glad that you did decide to join us. Now, I don't know about you, but when I hear the word hibernation, I do think of bears curled up in their warm, cozy dens, sleeping away until it's warm enough to wake up and find something to eat. Well, I think that's a pretty smart animal, if you ask me, and a lucky one. Why can't humans do that? Well, our very special guest this afternoon, Dr. Evangelos Michelakis, has a surprising answer to that question. In fact, the answer is the basis of his research that could transform the world of transplantation as we know it today. Now, I should add that Dr. Michelakis is joining us from Greece, where I believe the time change puts him into the wee hours of the morning. So we're even more grateful that he is here with us today. Now, to guide us through this fascinating journey, I would like to introduce to you Dr. Jody Abbott, our President and CEO at the University Hospital Foundation. Now, Jody, before we begin, have you ever thought about how nice it would be to hibernate all the way through our freezing cold winters?
Oh, Carolyn, I think I'd get a little bit bored from that much inactivity, but certainly the occasional weekend of being curled up would be a great thing. Good afternoon, everyone. Thank you so much for joining us. As Carolyn alluded to, today's virtual expedition will likely get you thinking about things you have not thought about before. Now, some of you may have recalled hearing or reading about Ignite 2030, our strategic plan for the next decade. The promise we make in Ignite 2030 is to continuously seek and inspire and lead bold solutions to really insurmountable challenges and ignite new discoveries in health. Well, while we weren't really thinking about Dr. Michelakis' research on inducing hibernation in human organs when we developed the promise, I think we might as well have been, because it fits really well. The gap between supply and demand of usable human organs for transplantation has been a major healthcare issue for years, and not just in Alberta, but around the world. Brilliant medical innovations by two of our own, Drs. Jayan Nagendran and Dr. Darren Freed, who were featured in a virtual expedition last February, have brought hope to people waiting for a lung transplant and have saved the lives of several patients right here at the University of Alberta Hospital. But transplant wait lists for all organs are still too long. There are over 4,000 Canadians waiting to receive a transplant as we speak. Their wait time can be from 2 to 7 years or longer. And on average, more than 200 Canadians die each year before getting their second chance to live. Unfortunately, these numbers are not decreasing. The research you're going to learn about today aims to eliminate organ transplant wait lists once and for all. In fact, it's about creating a surplus of organs and storing them in organ banks, similar to blood banks that have been commonplace in our society for decades. The impact of organ banks would have on the world of transplantation is simply astonishing. Imagine scheduling a surgery for a kidney transplant as easily as you might, for example, for a hip replacement. You would have more control over the timing of the surgery by booking it in advance. You'd have time to prepare yourself physically, mentally, and emotionally. If you were from out of town, you'd have time to make proper travel arrangements and ensure that the family you're leaving behind, including young children, would be cared for. And from a medical perspective, you'd be receiving the perfect organ match for your body, as opposed to the best donated kidney available. And most importantly, your prospect for a successful transplant and healthy recovery would be vastly improved. In other words, the creation of organ banks would transform transplant surgeries from unscheduled, traumatic, and nerve-wracking events to a practice known as precision medicine. Precision medicine because it's a medical procedure that is precise to your needs, your body, and your life. Now I'm going to say something that will really floor you. The research that Dr. Michelakis and his team are doing really goes beyond transplantation. As you'll hear today, inducing hibernation in human organs could also impact emergency care for patients having severe heart attacks or strokes. It could have significant influence on the future of space travel. And as some of you may already be thinking, developing the ability to effectively put our organs to sleep for extended periods of time could add years to the life and health span of the human race. If that strikes you as research worth knowing about and possibly even supporting, you're in the right place. It's now my privilege to introduce you to our special guest. Dr. Evangelos Michelakis is a professor and associate chair of research for the Department of Medicine at the University of Alberta's Faculty of Medicine and Dentistry. He's a clinical scientist at the University of Alberta Hospital and a fellow of the Canadian Academy for Health Sciences. Something else about Dr. Michelakis that you will find interesting is that his previous research on cancer received coverage from the New York Times, The Economist, Newsweek, and he's even been on CNN. So please don't hold back on any questions you may have. Dr. Michelakis, thank you very much for joining us from Greece.
Thank you very much. And thank you all for coming to this talk, and the foundation for supporting my work. I'm going to ask you to imagine today and imagine the future. And Albert Einstein said that imagination is everything. He believed that this is the most important skill of a scientist. And he also said that if you can't explain it in a 6-year-old, it means you don't really understand it yourself. And going through this exercise of trying to explain to you one of perhaps medicine's and biology's most complicated areas humbled me and actually made me even think even harder to understand some of these areas. The dogma that medical students learn in medical school, and the dogma that all doctors believe throughout the world, is that tissues can sustain lack of oxygen and nutrients only for minutes before irreversible damage occurs. So if you have a blood vessel in your heart and it's blocked during a heart attack, you only have 30 minutes or less before irreversible damage occurs. And if this is a blood vessel going to your brain, you even have less time before damage occurs. And as you can imagine, an organ can survive out of the body, in other words, with no oxygen and no blood, for up to a few hours, but during that time, it sustains significant and ongoing damage. While the size of transplant lists, as you heard from Dr. Abbott, keeps increasing, the majority of donated organs are discarded. For example, 70% of donated organ lungs are discarded because they're either explanted far from the recipient and they undergo ongoing damage while they're being transported back to the hospital where the patient waits because of lack of oxygen and nutrients. Remarkably, this rapidly growing and expensive field of medicine relies on ice buckets that you see here in transportation of donated organs. Experimental and ex vivo perfusion protocols, like the one that you heard from Dr. Abbott performed here in our hospital by Drs. Fried and Nagendran, for some organs, can extend the shelf life of the organs by a few hours and attempt to repair some of the ischemic damage that took place during the transportation. But they're utilized in less than 10% of transplanted organs. Now, I will ask you to imagine 3 things. First, imagine injecting a chemical in a donated organ and making it resistant to the lack of oxygen and nutrients, keeping it healthy in a simple refrigerator, not even a freezer. And therefore, also imagine a bank of donated organs in a refrigerator, similar to a blood bank where a transplant surgeon can pick the best match for a patient in need and place and perform the transplant surgery electively and not urgently in the middle of the night. You can understand that if this is true, there will be a very large increase in organ utilization, as I mentioned, a huge decrease in patient and family stress, big cost savings, and most important of all, much better outcomes since the recipient would be matched to the best possible organ. And most of the complications and bad outcomes in transplant surgery have to do with the fact that the donor and the recipient were not precisely matched because the recipient was in need of, and the doctors were desperate and they had to match a not perfectly matched organ to perform the surgery. Now, also imagine the second thing, injecting a chemical in a critically ill patient suffering from a heart attack or a stroke or multi-trauma or sepsis, like our well-known COVID patients, making them resistant to the lack of oxygen and nutrients temporarily until recovery takes place, meaning you may be able to skip intubation and allow more time for corrective procedures or simply time for healing. And now the third thing to imagine is the ability for deep space travel that this procedure would allow us to perform, since astronauts and passengers could be placed in a hibernation and wake up healthy on destination, like a bear waking up healthy after months of winter hibernation. Is this possible? When we have challenging questions like that, we often have to look into nature and we'll find an answer. And if you look into nature, you will see the example of a bear that is hibernating, and you will notice that the heart rate of a bear in hibernation is less than 5 per minute out of more than 150. And the respirations are less than 1 a minute, meaning there is minimal supply, if at all, of oxygen and blood in the big body of the bear. In addition, when the bear wakes up, right away, she's able to run and will actually chase you if you go poke her, as if hibernation never happened. But humans, when they go in an ICU or they are bedridden for more than a week or two, they undergo significant bone loss and muscle loss and inflammation in their body and all sort of other things that obviously the bear doesn't go through. There has to be a magical program that coordinates a number of parameters that give independence from fuel and nutrients, and also prevent any tissue deterioration during this prolonged immobility. Now, you may be thinking of polar bears when you think of hibernation, but many animals actually undergo hibernation even in warm and dry climates, from insects to birds to squirrels to hedgehogs. And again, it doesn't have to be because it's cold. Very or extremely hot temperatures, like in these examples of these reptiles, push animals to go into hibernation as well. There are some animals, like this toad, that when the dry season comes in warm climates, digs a very deep hole and can stay in hibernation for up to 9 months before waking up again when the rain season comes to play and mate and live happy. So when we look at the animal kingdom and our evolution, we will see that a number of animals from all stages of evolution can actually hibernate. And you would probably correct if you guess that although humans, ancestors, used to hibernate, when the humans took control of their environment and were able to make clothes and store food and migrate and build houses, the need for hibernation was not as pressing. So we sort of parked it on the side. We believe that the machinery to ignite hibernation exists in our systems, in our body. It had just been suppressed, perhaps temporarily. To learn about these mechanisms, we go all the way back to the origin of life. For example, this porifera, the species from which everything arose. These are simply sea sponges. And this took place more than 700 million years ago. Now, what was happening at this time? Earth was a very harsh environment with a lot of radiation and temperature changes and lack of nutrients. A lot of complex organic molecules have not occurred. So when the first bacteria came to life, they had to first develop mechanisms to protect and repair against this extreme environmental stresses and the lack of proper nutrients. It was not until the climate on Earth stabilized and improved and the ozone layer formed and there was no radiation and oxygen became available that animals started developing ways to grow better and reproduce, but retain the ability to go back to this early, primitive, but very essential forms of stress defense when nutrients and other stresses take place. Now, the way to do that, you need to have a sensor that tells you when this is about to happen. And at least when it comes to oxygen and nutrients, this job is done by mitochondria. Mitochondria used to be bacteria that at some point they infected the cells as we know them today. And both the invaders and the host like each other. The invaders, because they took advantage of the cellular machinery for a number of jobs they had to do on their own. And the host, because mitochondria, those early bacteria, had a superb way of generating energy. So they liked each other and they lived happily ever after in a symbiotic relationship. But mitochondria do much more than generate energy, being the powerhouses of the cell. They can actually sense the supply of nutrients and oxygen and be able to match the demand for fuel, oxygen, and nutrients to the production of energy. In other words, to the supply of energy. And remarkably, if the production of energy by mitochondria can match the demand, then life goes on. But if there is a mismatch and the cell starts to malfunction, including the cell's genes, the mitochondria have the job to actually make the cell die and commit a form of suicide called apoptosis. And the reason that this is done is that because the cell do not want to pass to their progeny defective genetic material. So they kill their host. Now, going back to these early primitive stresses, science discovered a number of mechanisms and genes that we now know them as longevity genes. And these longevity genes are called like that because they prolong life in animals and probably in humans. We just haven't quantified it very well yet. And these are genes that are activated when brief periods of stress that in biology we call them hormetic events. So these are hormetic response genes are taking place. And they're all factors that have to do with metabolism in mitochondria, ranging from sirtuins that are the most well-known and very well linked to prolonged life in a number of different animals. mTOR, this is the human target of rapamycin. That's a drug that used to be given to patients in the early transplant days. This is a molecule that is a sensor of amino acids. In other words, every time you have a steak, this molecule is activated. And if you don't have a steak, this molecule responds accordingly. And this last one called AMPK, which is a sensor for the supply of carbohydrates and lipids, the other major fuels in our body. This is a drug that is targeted by a very common drug used by diabetics, metformin. So how does this work when stress takes place? In this case, calorie restriction. Calorie restriction is a hormetic response. Another way of looking at that is intermittent fasting, which is currently the most popular form of diet throughout the world. That means spending at least 16 hours of your day without eating and only having one meal a day, perhaps in a 4-hour period around the evening. Now we know how both calorie restriction and intermittent fasting works because they seem to activate these primitive stress responses that are in all cells, involving molecules such as the ones I told you. And when these molecules are activated, they promote autophagy. That's a major repair process in the cell, where if nutrients are missing, then the cell, instead of waiting to build new ones from food, it actually recycles its own organs and reassembles the pieces and makes new organs by itself. You can make new mitochondria. You can protect the integrity of your genome, like your DNA code, and overall become stress resistant. And you can understand why, if people are able to activate these pathways and are resistant to these stresses, including disease stresses, they live longer. That's why we call them longevity genes. And activation of these parameters has been clearly shown to lead to less metabolic disease and diabetes, less cardiovascular disease, neurologic disease and dementias, and overall improve longevity. How is this very complex response to stress gets coordinated, particularly when the time comes, a time of need comes? Well, like I said, it is in our DNA, like what we think the hibernation genes are, but they're not needed all the time. They're only needed, these mechanisms, only when stress comes, like when hibernation is needed. So this is coordinated by something that in biology we call epigenetics, from the Greek word epi, which means on top, and genetics, because these are signals that are sitting on top of your genetic code. If you think of our genetic code like the hardware of a computer that we all have, epigenetics is the software on these computers that allow one computer to run this program at that time and another computer run another program at another time, and so forth. Just think of this for a little bit. When we're born, our early embryo, like you see here, this original cell from a man and a woman merged and started dividing, and then here we have 8 cells, and from this you're going to get a whole body as it grows. But you're going to get different things like blood cells and nerve cells and heart cells and skin cells and bone, very different to each other cell types. Yet all of them have exactly the same genetic material, exactly the same genetic code. So how does this happen? It happens as I'm going to show you, but before I go to my next slide, I will mention here that when this process of giving different identities to different cell types actually goes wrong, you get cancer. So the implications of these pathways are important from cancer, from how we are normally evolved, and multiple aspects of disease, and even aging itself. Now, I want you to, before I tell you how this is done epigenetically, I want you to look at this video that shows you the double helix, your DNA, the big discovery in the 60s that said that this is a double helix of the genetic code, as I'm sure you've heard about it. Now, how is this packaged in cells? It is packaged because some proteins called histones that you see here in blue come and attach on the double helix and then force it to wrap around them, forming what is known as nucleosomes. And these nucleosomes can actually get attached to each other and form a super pack, so to say, so that a very long chain of DNA, that if you were to get it out of a single cell, it will be approximately 4.5 feet. So the DNA in a single cell is 4.5 feet. But yet it needs to be packaged into the nucleus, that little tiny part in the cell's body. And just to give you an idea how small we're talking, this many feet of DNA has to be in a nucleus, and it takes 10,000 nuclei to fit a tip of a needle. Now, if you zoom out, you'll see that this chromatin, or this DNA and histones, is actually what chromosomes are made from in a living cell that you can see here that is dividing. There is a one daughter cell and the other daughter cell, the chromosomes split, and that's life as we know it. Now, how does epigenetic regulation occurs? You can imagine that if you're so tightly packed, it would be very difficult to read the genetic code. You have to unpack this tightly packed chromatin and the histones to allow the readers of our genetic code to actually read some specific genes in specific parts of a chromosome at specific times. So here is again the DNA helix, and this is the histone, and the DNA is wrapped around the histone. And we now know that simple addition of organic molecules like a methyl group or an acetyl group can significantly alter the biophysical properties of the histones, allowing them to relax that superpacking and enabling the readers of the genetic code to read specific genes at specific parts of the chromosome on demand. And this is organized in a way that when the mitochondria sense that there is a high nutrient condition, there is a lot of histone acetylation, for example, and the histones are loosened up and able to allow the readers to read our genetic code and make our proteins as we know them, versus low nutrient conditions that are associated with low acetylations of histones, for example. And you can see here the other mitochondria-related molecules, the AMPK that is here, or the sirtuins that are into this place here. So we believe that something like that could be happening in hibernation, like the genetic code has the hardware ready, but our software has actually asked those genes to be silenced and stay quiet because they're not needed, and there's no need to activate these mechanisms. If we were to allow it to be activated again, we may be able to hibernate ourselves. Now, I want to tell you something more specific about mitochondria and epigenetics. Mitochondria is the field I'm working on, and mitochondria can regulate many genes epigenetically, like the histone acetylation, as I told you. And this is some very recent knowledge that we published in a prestigious journal back in 2014, showing that an enzyme that produces acetyl-CoA into the mitochondria, and acetyl-CoA is the molecule from which you get these acetyl groups to get histone acetylation, this little organic molecule that was attached in histones, can actually approach the nucleus of the cell as if they were bacteria, because they used to be bacteria, sort of attach to it and inject in it one of the biggest enzymes they have, is called pyruvate dehydrogenase complex, which is the complex that makes acetyl-CoA, and then directly affect the histone acetylation in very large amounts. Just a couple of months ago, we published another paper that we described the mechanism for doing so. And we showed that mitochondria can actually crawl around the nucleus and attach to them, to the nucleus that you see here. And this is a mitochondrion, that's another mitochondrion, attached with these tethering proteins called mitofusins, and form a stability through which they can inject that big enzyme, which you see here in a dividing cell. You see the two daughter cells. There is a nucleus, one half of the nucleus, the other half of the nucleus, and the DNA and histones are in blue, whereas this enzyme that produces acetyl-CoA is in yellow. And you see when they donate it, it just goes and wraps around this part of the DNA, providing acetylation acetyl groups to acetylate histones, change their function, and allow a number of genes to be activated. How would we study these possibilities, these potential mechanisms in nature? We chose to study snails because there are plenty in Alberta. They hibernate. They don't hibernate just in heat or cold. They can hibernate anytime in our laboratory, for example, for as long as there is a dry period. Like if the air is dry, and you don't give them water, they hibernate. They form this membrane, and therefore you can easily tell that they're hibernating. And we can study them. We can get their blood, we can transfuse them. They may look very different, but they're very similar to a human being. They have a brain, and they have a heart, and they have lungs, and they have blood vessels, and so forth. So we believe that they sense the changes in the environmental conditions in the brain. They secrete something that we call hibernation inducing factor to the whole body, and induce epigenetically this whole change is to prepare the body to withstand lack of oxygen and food and everything that I talked about. We study them with a collaborator that is a researcher in cardiology and cancer, Gopi Sutendra, and Patrick Huntington, a scientist at the U of A School of Public Health, who studies snails. Snail biology, and we have them in the lab, and we put them in the dry air area or the refrigerator, and when they wake up, they start moving right away. You can see the snail here, whereas others haven't woken up yet. They're about to, and they remain sealed. It's fun to think how French chefs cook snails. They love it because they can keep the snails in the refrigerator, take them out the night before. The snails wake up, they're perfectly healthy, although they've been in the refrigerator for months, and they make their dishes the following day. So what we can do is to actually put them on a flat surface and put cameras on top, and we can tag them and track their activity. So we can see which snail walks around and in what speed and exactly when. So we wanted to see if this substance, the hibernation inducing factor that is coming out of the brain, and it makes the whole body withstand this lack of nutrient stress, is transferable because it's circulating in their blood. Can we take it out? And so we did blood transfusions in snails, exactly like you do them in humans. And we got a sleeping snail, like a hibernating snail, and we gave its blood in a control snail. And when we gave the blood in the control snail that was awake, we gave it blood from a sleeping snail. The snail started getting into hibernation within a matter of just a couple of hours. You can see the activity, the distance, and the speed in which they travel slowly went down and became inactive. But it didn't die, because a day later, when perhaps the effects of the injection were removed, the snail recovered, as you can see here in blue. And if you look at our mean data, these are a number of snails that are control, and they got blood from another control snail, an awake snail. Nothing happened in their activity. But if the control snails got blood from a hibernating snail, you see they got a significant suppression of their activity, and they became hibernating snails, exactly like the real hibernation snails that became like that because the environment became dry and they didn't have any water. These ones did have water, but they just got an injection of another hibernating snail. So this showed that this hibernation-inducing factor is transferable across different snails. That was a big discovery, we felt, when it hasn't been shown before in other animals. And then when you study the snail tissues that are either hibernating from, tissues from hibernating snails, or tissues from non-hibernating snails that received blood from a hibernating snail, the transfused snails, you see that the oxygen consumption, in other words, their mitochondria function, because that's what mitochondria do. They consume oxygen to make energy out of that. Their mitochondria function was suppressed. And then we wanted to see if the same way HIF is transferable from one snail to the other, can it be transferable from snails to other species like human cells or rodent cells? So we took the blood from a hibernating snail and we gave it in cells from rat and human lungs that we isolated. We plated them on a Petri dish, and we gave them the blood from a hibernating versus a normal snail. And the first thing we showed is that these cells, whether they're human or rat, doesn't matter, it happens in both, they have much increased acetylated histones. This is the epigenetic phenomenon I was telling you. So this is what happens if in the hibernating snail, this is in a snail that gets an injection from a normal snail, but this is what happens from here when he gets the injection from a hibernating snail. Histone acetylation increases significantly. And you can see that with a different method here. Under a microscope, you see this blue, this is the acetylated histone. It's much more after you give a hibernation blood compared to blood from a non-hibernating snail. And these are human cells. In fact, here you have pieces of lung and pieces of pulmonary arteries. These are the lung blood vessels. And if you put them in a Petri dish and you bath them into the hibernating blood from the snail, they get a significant suppression of mitochondrial function, whether they are pieces of tissue or pieces of blood vessels. And a day later, this comes back. It's not that they're damaged, they're reversibly suppressed, like it happens in the snails. So the value of that is now if we give this hibernating blood from snails into human cells that are exposed to very low glucose or low oxygens, in other words, harsh, low nutrient conditions, the same way that a explanted human organ would experience, we see that these cells can handle it much better. And P53 is a master molecule that regulates this mitochondria-driven apoptotic death, the kind of death that kills the cell when fuel doesn't match with fuel demand, doesn't match with supply. Remember that concept? So you see here much increase in the P53 levels when you give control snail blood in fuel-starving human cells. But if in those fuel-starving human cells, you give them hibernation blood from snails, there is much less P53. And there are many other ways that you can measure cell death and handling of stress. And of course, they are better. If you think of this, there's a circulating substance, you may ask yourself how we're going to make a medicine out of that. Well, first, we need to find exactly what this substance circulating in the blood of a hibernating snail is. And these are fancy techniques, NMR and mass spectrometry, that we can compare the blood from a hibernating versus non-hibernating snails, see the differences, and chemically analyze them. And we're now in the process of chemically synthesizing that hibernation-inducing factor. That we can perhaps then give it to a human organ. We get this lungs out of patients before the transplant, and they're connected in a ventilator through the bronchi and in a pump circulating blood or other solutions in the pulmonary arteries. And you can see it here. This is the work done from Dr. Najendran and Fried. And you can see this human lung that was donated for transplant, but was deemed to be not optimal for transplantation because of damage they have suffered during the transport. So Dr. Najendran and Fried put it in this system that perfuse it, sort of clean up the inflammation, and they resuscitate it. And they can actually keep a lung for many hours in that system following oxygenation, swelling, stiffness, pressure in the lungs, all factors that have to do with quality of the organ function. And last year, they had a prestigious journal publish their work that they were able to rescue 12 out of 12 of these suboptimal organs that would normally have been disregarded, but actually they corrected them within a couple of hours and they were able to transplant them to the patient successfully. So obviously we want to be giving this hibernation-inducing factor in these human organs and measure their performance the way I showed you over days or weeks perhaps, or store them in the refrigerator and then take them out 1 or 2 weeks or 2 months, who knows yet, we don't know, later, and hopefully show that they have good performance. In my last slide, I'm gonna tell you that we did this work with funding from the New Frontiers, which is a federal organization funding a high risk, high reward, in other words, provocative projects like ours. And we're close to finishing this milestone, but then we need to move into number 2 milestone, elucidating the effects of HIF on human cells and organs. It is then that we're going to apply to CIHR and NASA. As I said, no, I didn't tell you, but I'm telling you that I've been funded from CIHR for many, many years. And I can tell you it is very hard to get funded with a project like this, because it's sort of kind of unbelievable, and scientists are by nature very skeptical. You need to show much more progress before NASA and CIHR can actually fund you further. And this is why Hostel Foundation is very critical in supporting projects like this, high risk, high reward projects at their vulnerable state, before we can actually perform clinical trials with these organs exposed to this hibernation factor and being transplanted out of the refrigerator to humans. I wanted to thank you all for listening, and I appreciate your presence and the Hostel Foundation support of my work. Let me come out of the screen share. There you go.
Thank you, Dr. Michelakis, for that fascinating presentation. Like everyone here, I'm really looking forward to hearing more during our Q&A. My name is Shannon Diaz, and I'm a member of the development team here at the University Hospital Foundation. I'll be the moderator for today's question and answer period. If you have any questions, you can submit them with the Q&A button here at the bottom of the screen, and we've received a few already, so let's get started. Our first question: is it possible to induce hibernation in our entire body or just the organs?
Yes, of course. As I implied, if you inject a substance, like whatever substance that is in the body of a snail or a bear, it does put the whole body into sleep. It's just that some organs are more demanding and difficult than others, like the brain, for example, compared to the lungs. We chose the lung first because the lung doesn't consume a lot of oxygen. Paradoxically, it's designed to supply oxygen to the body, but itself, it doesn't consume much. So it appears that it may be one of the easiest. That's why we're starting from a lung. But other than that, our next steps would be in all sort of organs and the whole body itself.
Okay, great. You just answered the second question, which is about whether all organs respond the same. So that's great. Thank you. Here, I've got a fairly long one here. Andrew is saying, thank you for the stupendous presentation and illustration of the most up-to-date novel translational medicine research conducted to benefit the transplantation patients. His question is, the placenta as a dynamic organ works as a perfect shield to protect the fetus from a plethora of immune responses. Would studying the placenta cell traits, mitochondrial proteins, and surface receptors shed some light on conditioning the donor organ's vascular endothelial mitochondrial program to withstand host immune responses without the need to maximizing the current immune suppression regimens on the long run, i.e. studying the dynamic placental mitochondria at various time points? Hope I got that okay.
That's a very good question. In fact, we have studied placental mitochondria from humans and different animals. I used to do that many years ago, because the placenta is like the lung, but for the fetus while in the womb. So that's what it is. And it has very similar functions. And the same way, the same principles that I explained in how the mitochondria sense nutrients and coordinate this process. Something similar happens to the placenta. It's just that it's much easier to get human placentas, and I think that's what the question was going, than lungs. But lungs is where the immediate need is in medicine, and that's why we're starting from this harder part. But I think that if we were to collect placentas and do similar work, we wouldn't be that far from the lungs. They're very similar.
Excellent. Can you tell us what role inducing hibernation in human organ plays in trauma care?
So the same way, if you get multi-trauma, a lot of organs are damaged, and often your heart, and you're bleeding, and therefore you're losing blood, and you don't have much oxygen. And of course, you need them for the healing, for the survival and the healing. So often the patients die because of the lack of oxygen and the lack of blood, or massive amount of blood loss, before the surgeon can do much, or before the body's healing process can take place. If you were to inject a hibernation inducing factor in the ambulance that transfers a multi-trauma patient, by the time the patient arrives in the emergency room and the surgeons start taking care of him or her, then his or her body would be entering hibernation, would no longer be in as much need for oxygen and nutrients. And like the bear that can live with the 5 heartbeats or 1 breath a minute or less, the patient may survive. And like the bear that can stay healthy, muscles and bones, while in the ICU for in the cave. The same way the patient, several weeks, like this multi-trauma patient spent in the ICU, can actually come out of it without having lost muscle and bone and many other things.
Excellent. All right, our next one is a bit of a comment, and I wonder if you might be able to touch on this. The comment is, this is very interesting. The science is important, but I wonder if it will be useful for transplants, because I thought most transplants are used and hence wouldn't be available for storage to allow better selecting of donors and recipients. So I think this touches on what you were talking about earlier about the wastage of organs.
It's very unfortunate, but the fact is that most of the organs are not actually, the organs offered are unfortunately not used because of the reasons I explained. They just get damaged, and they by the time they arrive, they're actually in a bad shape to be put in a desperate human being. So they're not used, unfortunately.
All right, thank you. Hopefully we'll help solve that. We're almost end of time, but if you have a minute, we maybe ask 1 or 2 more questions. Is that all right?
Yeah.
Okay. How far are we from actually seeing organ banks at the hospital?
I think we need a bit of luck, but if you think about it, right now we have blood banks. We have also corneal tissue banks. People that need corneal transplants go into a bank like that and pick, because the cornea doesn't have blood vessels and doesn't need oxygen and nutrients, so you can store it the same way that we could be storing human organs. So in that timetable that I said, within 3 years, we may be able to actually start showing if we have clinically relevant effects in perfused human lungs out of the body. And if we do, then a clinical trial will happen and use some lungs that are not normally used, injected with hibernation factor. And then if they do well, 6 months after being put in the patients, we will have the results. Typically, you say if everything goes well and you're very lucky, maybe in 5 years. But usually these are too optimistic, and the reality is that it takes longer, even in the best of circumstances. But about that kind of time.
All right, thank you. I see another question here. Are you closer to understanding how to minimize the tissue degeneration issue?
We actually do know a lot about that. We know how the tissue degenerates. We just don't know how it regenerates easily. So there are a lot of parameters that we can measure in the blood or in little samples on the tissue that show the decay of function and all the abnormalities. You can take samples and immediately get your answer biochemically or under the microscope, and of course by functional assays on how the tissue performs. So all these parameters that I showed, how stiff the lung is, how the oxygenation efficiency takes place, how high the blood pressure is in the lungs, how leaky are the blood vessels in the lungs. All of these things are indices of tissue decay, and they can be supplemented by more sophisticated things by checking chemicals that are released by damaged cells and looking at pieces of tissue under the microscope. So I think we're very good about that. It's we just need to know how to prevent that.
Great. All right. I think we'll do last question here. Is forcing hibernation like inducing a coma?
Kinda. Just when you're in a coma, your brain is inactive, but your brain still needs oxygen and blood, as the rest of your body. That's why people in a coma may need to be on a ventilator or will need transfusion of nutrients or tubes in the stomach to feed them. So they do not become independent of fuel, independent of oxygen and blood. So the activity is down in the brain, but the dependence of the body on these nutrients remains the same. It's actually exactly the same as that of a healthy patient. So the question is how to suppress the need for fuel in the rest of the body in a sort of a similar way like the function is suppressed in the brain.
Well, thank you so much, everyone, for all of your questions. And that is all the time that we have for those questions. And all I can say is, wow, what a journey we've been on. I am just in awe of these advancements being made in transplantation and expect many of our viewers are as well. On behalf of the University Hospital Foundation, including Dr. Jody Abbott, Shannon, Emily, and myself, and of course, our very special guest, Dr. Evangelos Michelakis, thank you for your time and active participation. And I should mention to everybody that it is 2 a.m. in Greece for our good doctors. So thank you even more for joining us. Within the next few days, we will send you a very short survey for you to let us know how we did today. We will also be sending you the recording from today's expedition. Now, if you want to learn more about us, the University Hospital Foundation, please reach out by email or visit our website at give2uhf.ca. You can also watch recordings from our past virtual expeditions on the University Hospital Foundation's YouTube channel. And we also hope you will join us for our next expedition, which will be held on Tuesday, June 7th. And the topic will be the rise and fall of diabetes. So as we mentioned earlier, ongoing community support is vital to ensuring the work of our innovators. We want to make sure that we can continue to make breakthroughs and advance patient care and improve the health in our communities we serve and around the world. So thank you for joining us today. And this concludes our formal presentation. Before we sign off, we'll leave you with a very short video entitled Believe It. Thanks so much, everybody. Have a wonderful evening.